Oh, yeah. It's a pleasure to be here, and it was a kind of a nice, pleasant surprise that the NSF organized, well, NSF funded the organization of this, but I think they also put a lot of intellectual content into the exact details of how it was done. I have a sort of strange relationship with aggregation that goes back quite a while. In the early 90s, I was a Fogarty Scholar in residence at the NIH, the intramural program, and I thought I would take advantage of the fact that Bill Eaton was there. He had been the person who invited me, who worked out how aggregation is involved in sickle cell disease. And of course, I'd heard about prions as being involved with, at that time, being described as misfolded proteins, and I'd been doing things in folding. And so I decided to spend about as much as I could of that year focusing on the prion problem. And I would read paper after paper after paper. Every paper I read, I would conclude that control had been left out of the previous paper, and then that would be corrected in the next paper by Prusiner, and then that would be corrected by the next paper. And then finally, just about the time I was about to end the year, two things happened. A paper came out in Science that said, well, they actually found DNA in a prion disease. And then I said, well, that's the end of it. I'm not going to study aggregation anymore. Of course, then in the fall, Prusiner won a Nobel Prize, and so now we knew it was true that aggregation was the cause of prionic diseases. So I sort of avoided the problem for a long time, and then I think it's maybe about 10 years ago when I first came to Rice, I started to look at an experiment, my group actually started to look at an experiment of Jane Clark on the aggregation of oligomers. And she had explained it in a language that was consistent with some of the things we had been saying about domain swapping. And we started to study it, and we discovered instead that there were these amyloidogenic fragments that were causing a completely different mechanism of aggregation. And of course, I knew that was a ridiculous idea because David had had this idea, Dave Eisenberg, before, but I was slowly convinced by the data that he was absolutely right and that there really was a new phenomenon going on there. So I have a strange relationship with aggregation, and I've gotten into it, but in somewhat indirect ways. That investigation led me to do some things on A-beta, which I'm not going to tell you about. But a sort of different project arose in my group, which was that I'd heard again that there were prions were involved possibly in the mechanism of actual memory. This idea, I was hearing about it at the time, was due to experiments of Eric Kandel, Nobel laureate, and Susan Lindquist, who unfortunately passed away. And then I discovered, and so I asked my student, Ming Chen, to just look at the structure of the protein that they had identified, CPEB, as being involved in this process. And that's a sort of a standard starter project in my group, learn how to do structure prediction, which we've been doing for quite a while. And, okay. And so he looked at it, and I'm going to tell you first what arose out of that. It turned out that this problem of aggregates being involved in memory goes much further back than Kandel. At least we can trace it back to a paper by Francis Crick in 1984, where he states this thing which was called the memory timescale puzzle, which was that memories obviously last for 50 years sometimes for some of us, then they start to fade. And I'm not pointing to any particular person. And that time scale is much, much longer than the time scales that are involved in degradation and turnover of proteins. And Crick in his paper identified several ways that you could get around this. There could be something like an epigenetic switch that involves some sort of feedback cycle that doesn't use always the same proteins, but it has multiple protein concentrations. It could be based on phosphorylation, which he suggested. He also suggested that it could be caused by aggregates, which would last for very, very long periods of time, and you just swap in and out different members of it. So in fact, this idea of a prion as the origin of memory goes back to 1984 in Crick. So we pursued that. And one of the things that we found in doing the the simulations of that system doing the structure prediction is that that protein, CPEB, or at least the part that was thought to be involved in aggregation, didn't aggregate, at least in a trivial way, immediately to a prion-like structure at all. Instead, it was robustly predicted to be a helical bundle, which formed oligomers that were helical clusters. So this, I guess we would have stopped there and just had an amusing starter project. But Ming Chen said, you know, well, anything that's got helices in it, the helix is shorter than a beta strand, which we all learned. So if you could pull on it, then of course you could convert anything that's helical into something, if you pull on it enough, that's some sort of beta adduct. And so we had this idea, or actually Ming Chen had the idea, that this was an example of a prion that was caused by its interaction with some sort of pulling system. And indeed, this protein CPEB is co-deposited, when it's in this prionic form, co-deposits with other cytoskeletal proteins. So first of all, I think this is another example of what we were hearing about in the morning, that I think when we think about the formation of these aggregates in vivo, we probably are not studying exactly the same process that we study in vitro, and there may be detailed interactions with other partners. This actually leads to a kind of interesting story where there's another aspect of the crick mechanism involved. It turns out that this CPEB, this has been shown by Kandel, that the CPEB in the prionic form, in the beta strand-like form, turns out to be something that turns on the activation of other RNAs. It's a translational, what would he call it, accelerant? I forget always, it's the opposite of an inhibitor. Repressor, activator, yes. So it's a translational activator, specifically of proteins that are involved in the cytoskeleton. So you can see that once you form a little bit of this fiber, it will allow more actin to be made from F-actin and then that can make a transition from G-actin to F-actin, cause things to move around and stretch in some way. And in this way, one can have a feedback cycle that involves the formation of the prion and its activity as a translational activator. Let me go back. So in fact, this has led to a whole line of work that's been partially funded by the NSF and 3 papers on the CPEB3 prion, how it interacts with F-actin, where we've done experimental work with Neil Waxham and other people in our extended group. And then one of the things that comes about in this interaction is you go, well, why does forming an aggregate have anything whatsoever to do with turning on RNA translation? That doesn't sound like a very smart thing, or an obvious thing how that happens. And again, my student Xinyu Gu realized that the formation of a structure, like a prion, allowed you to have further processes to go on to have a structural and vectorial character. So she came up with the idea that we're calling vectorial channeling. I came up with the way to mathematically solve the transport problems that are involved in forming a small factory of a prion. And it turns out that it has an interesting prediction that depending on the orientation of where the binding site is the binding site for RNA closer to the fiber itself, or is it on the outside of the fiber? If it's closer to the outside of the fiber, you make something that activates translation. But if it's closer to the inside of the fiber, you have something that represses translation. And again, there are examples of prion forming proteins that are also RNA processing proteins, and that orientation dependence seems to be there. So first of all, I guess I'd say that when we think about aggregation problems, we should think about them not only in the context of disease, but in the context of doing good things like forming the memories in the first place. And of course, I think the fact that most of these proteins that we see are somehow involved in neurons, but we typically don't know their positive function, I think this makes us want to think in that way. Now, this sequence that does the formation of the prion has a lot of cues in it. So we also started to look at the problem of Huntington disease. And I think that the Huntington disease case, as Rohit was telling you, is probably the cleanest example of something where it's very clear that the physical chemistry of aggregation has at least something to do with it. Bill Eaton actually, at the time when I was visiting, we thought a little bit about Huntington's disease. He said it was clearly a disease of physical chemistry, like sickle cell disease. Now, the person who discovered that, made that clear, is shown here. It's Nancy Wexler. She's a really wonderful, wonderful person. I served with her on the NAS Committee on Science and Engineering Policy, so I got to know her quite well. She's a very soft-spoken but well-spoken person. She was trained as a psychologist, but she went into studying genetics because her family suffered from Huntington's disease. And she was able, by discovering a certain tribe that had a lot of Huntingtons in Venezuela, that the disease was indeed correlated to a particular protein, now called Huntingtin. She is part of a group. And also that its onset was clearly correlated with the physical chemistry of the repeat. I should say that just last year, I mean, I think it was maybe clear when I first knew her, she seemed quite somewhat a little bit frail person, but then in the last year she's sort of indicated that she does have Huntington's disease, which I think is ironical, or not ironic, I don't know what the word is, sad for me anyway. She's a very nice and very great scientist. So here's the data that comes out on that. This is the age of onset as a function of the length of the polyQ repeat. And you can see that if you have a repeat that's, say, 20 long, which most of you probably have, there's nothing for you to worry about. But as you penetrate into things you have lengths greater than 40, you start to get Huntington's disease at an older age. And then as it gets extremely short, as happens in these people in Venezuela, sadly, you then get a very early onset version of the disease, very fulminative disease. Now, so this actually overlaps with Rohit's talk, although we came at it from a different direction. When we do those same kind of simulations that we use to look at the prion, mechanical prion, when we apply this to polyQ, what we find is that depending on length, you have a sort of bias to being more compact tadpole or a more extended tadpole. And the shorter segments tend to be more favor the most extended form. But as the thing gets longer and longer, as you can imagine, if you have something that's going to stick to itself, it wants to fold back onto itself. So the distribution for something which is quite long is favoring something that's already partly aggregated. Because it's partly aggregated, it's more rigid, and that's why it now is able to bind to other copies of itself much more readily, in our view. So in fact, we're able to also add on the nice ingredients that Rohit talked about, the N-terminal fragment, which seems to encourage aggregation. It does that because it is, again, one of these helical bundles, and they start to come together, and now you've raised the local concentration. So a pure N-terminal version plus Q aggregates with a much shorter Q. That's in agreement with experiments by Wexler and others. And then on the other hand, there's the polyproline segment at the end, which works in the opposite way and discourages aggregation. And When you put them both together, you find that something with the length, say, of 20, the aggregation, and you can calculate the free energy as a function of the oligomer size, and concentration, you discover that at the concentrations you find in inclusion bodies, the short fragments are uphill and they won't aggregate. On the other hand, things with the longer fragments will in fact be downhill, and that's why one does start to form fibers in those inclusion bodies. So this is an example, I think, of a disease where physical chemistry certainly correlates with the phenotype that we observe. So with that, I'll stop just to say we're still quite interested in general with the problem of memory formation. This work was funded by the NSF, and these are the people involved in that, and Herbie was involved in it as well in a different part of the process. And this is our sources of funding. Thank you.
Thank you, Peter. So we have time for a couple questions. People are getting tired.
Yeah, Sheena, or well.
I wondered, you didn't really talk about this, Peter, but in terms of a general question about simulations and understanding aggregation, you talked about the phase transition changing, the probability being aggregated or not, but what about the mechanism of forming those aggregates and the steps that you have to go through? Can you calculate that?
Yes, I believe we can. We haven't, in the Huntington story, well, first of all, let me go back a little bit to pat myself on the back. Most simulations of aggregation simply say, oh, I saw the thing aggregate. And the reason is there's actually a technical problem, which is how do you convert a simulation in a finite-sized box with a finite number of oligomers into a free energy profile that depends on concentration. That those profiles depend on concentration, and it's always the grand canonical free energy. So it's actually a kind of simple thing to do to correct for it, but virtually no one did it. I think that the energy functions we use are, while coarse-grained, that lets you simulate big systems, are reasonably accurate. So we've been able, for example, in the A-beta case to predict things about the, again, the concentrations at which things go downhill, but also we made the interesting prediction that the growth involves undoing a frustrated interaction in the tip of the fiber, and that led to the prediction that the growth should be harder as you add urea, because urea, of course, denatures proteins, tries to remove them from aggregates. On the other hand, that it speeds up by the addition. So it's something with a -5 value for aggregation. So I think there are at least some features of the mechanisms that we're predicting and we're comparing with different experimental groups, but not so much on Huntington. I'm not quite sure exactly why we stopped working on that, but we got distracted with other proteins.
Peter, the physical chemistry of memory consolidation a la Kendall and Lindquist, it's a nice story. What about the biology? I haven't followed that. Is there more evidence?
There's more stuff going on there for sure. For example, this protein CPEB is also sumoylated, and that is a sort of a side issue. She, I guess is his name, and Kendall seem to still get results that are consistent with this mechanism. But in detail, I think we need to finally understand how you grab on the CPEB from both ends. And actually, so far, we've only studied the problem of how does it associate with it on one end, and that's just been because of time and money.
Okay, we have one more question, but why don't you get Garrett?
Yeah, sorry.
Computers while you answer one
Oh,
question.
maybe I should put that one up though. From Sidney Benner.
Oh, sorry.
He just says you actually have to think about molecules. Okay. Okay, great. Thank you.
One more question, Peter, while Garrett gets ready.
Quick,
Oh, sorry.
yeah, so we recently published a paper on the cryo-EM of Huntington.
Yes, I saw that. Yes, and yes.
But I was wondering in terms of these out of register and the stashing angles of the polycube. Do you pick up these when you're simulating?
Well, I only saw your paper fairly recently, unfortunately. So we didn't make a quantitative comparison, but indeed we do see lots of different orientations and things without being in complete registry in our simulations. This is related to something that I talked about once in the context of protein folding, something I call the polyethylene problem. If you think of polyethylene, obviously it turns out the big innovation in having good plastics, like that we can pollute the environment with, is to be able to make them crystalline. But the crystals of polyethylene are not like the crystals of anything else. There's things where things go out and come back in and so on. And we definitely see that with the polyglutamine, pure polyglutamine. With the Huntington, we didn't look at that yet. Thank you.
Okay, you're getting late, but you guys are going to have a